This session's for the Advanced Cell Biology module, and we're going to cover cellular regulation via the ubiquitin proteasome pathway. So in this session we're going to look at the proteasomes, and the proteasomes are organelles that are involved in targeted degradation of specific proteins. And we're going to also cover ubiquitin, and ubiquitin is the tag that is attached to specific proteins when they are destined to be degraded via the proteasomes. So we're going to look at the key functions of proteasomes and ubiquitin in controlling protein levels, and look at some of the pathways that are involved, so some of this particularly related to DNA damage and related to cell cycle control that allows cells to rapidly degrade proteins that are no longer needed. So firstly we're going to look at proteasomes as organelles, and they are large protein complexes, typically in the cytoplasm. Some of them can be in the nucleus, and they form this sort of barrel-shaped or tube-shaped organelle, and their job is to pick up substrates, which are proteins which have been ubiquitinated in a very particular way, and degrade those proteins. Now the proteasome organelle itself is sort of a hollow tube-shaped organelle, and the proteins are threaded down this proteasome, and within there are three proteolytic catalytic domains which chop the protein up at specific amino acid residues and degrade the proteins into short peptide fragments. Therefore, if you can target a protein to the proteasome by having it tagged with ubiquitin, it will have a ubiquitin receptor here. That then will have a deubiquitination step, and that will strip off the ubiquitin proteins to leave the free protein to be threaded down the proteasome, allowing it to be degraded. Now this slide is showing the structure of the proteasome, showing the inner part of the 20S core, which has the catalytic chamber. And this core part of the proteasome is made up of all of these individual proteasomal subunits. So these are individual proteins making up each of these four rings, alpha and beta rings. And this is showing a sort of a cross-section through the proteasome, and each of these in green and yellow are the catalytic domains. So as the protein comes down here, there are catalytic domains made up from either these alpha or these beta subunits or these alpha subunits here to create an enzymatic catalytic domain to chop up the protein. So these are the chymotrypsin-like, trypsin-like, and post-glutamyl peptide hydrolase-like. So chymotrypsin, trypsin, they cleave proteins at very specific amino acid motifs, and post-glutamyl, as the name suggests, cleaves after a glutamic residue. Now the degradation of proteins is controlled by the protein ubiquitin. So when I talk about proteins being tagged with ubiquitin, they are being tagged by a protein called ubiquitin. This protein is a 76 amino acid protein, highly conserved across all living systems, and it is used in many different functions. So many proteins are tagged with ubiquitin. There is a particular type of ubiquitin tag that I'll come on to in a moment, specifically for proteasomal degradation. The protein ubiquitin is called ubiquitin because when it was first discovered it was bound to virtually everything. So it's almost like ubiquitously bound to things, and that's because of its role of being used to tag proteins for specific functions. There's as noted here, there's lots of other ubiquitin tagging systems that are unrelated to the proteasome. We're just going to cover one of those. For the ubiquitin proteasome system, you have a polyubiquitin chain of four or more ubiquitin proteins tagged to a target protein. So this slide starts to illustrate how the ubiquitin proteasome system is controlled and how we get ubiquitination of target proteins. So ubiquitin is covalently attached to a substrate protein, and it's a three-step mechanism. There is the ubiquitin activating enzyme E1 here, And what this does is binds to some ubiquitin protein. This is then passed over to the ubiquitin converting enzyme E2. So E1 has bound to ubiquitin and is holding on to it. E1 can bind to E2 and pass over that ubiquitin protein to E2. And then the E3 enzyme is the ubiquitin ligase, and it's the E3 which is crucial for tagging or binding to a specific target and allowing the passing of this ubiquitin to the E3 bound target. So there are very few E1 type enzymes. There are a modest number of E2 enzymes, but there are many hundreds of individual E3 enzymes, all with quite limited substrate specificity. And we'll have come across some E3 ubiquitin ligases in previous lectures, which I will mention shortly. So this is just showing the same information in a slightly different diagram. So here's the E1 binding to ubiquitin to create an E1 bound ubiquitin complex. This is passed over to E2. We get interaction with the E3 ubiquitin ligase, which is going to bind to a substrate, and then we'll get ubiquitination of that substrate protein, and we end up with a polyubiquitin chain. So E1 and E2 are crucial for activation of ubiquitin. E2 is crucial for passing over that ubiquitin to the target protein, but E3 must be present and binding to that very specific target. So it's E3 that makes this target specific. Now the E3 ligase that you will have come across is MDM2. You should be well aware of MDM2 as the E3 ubiquitin ligase that degrades p53. So in the p53 degradation pathway, if p53 is unphosphorylated, MDM2 can bind to p53. So this could be MDM2, this could be p53. This is unphosphorylated region of p53 because it's not been activated. That means it can be degraded. And in that situation, MDM2 can bind to an E2 ubiquitin bound protein, at the same time bind to p53, and then the E3 ligase can facilitate passing over that ubiquitin to p53, and we end up with p53 with a polyubiquitin tag. That p53 protein is then makes its way to the proteasome. The ubiquitins are stripped off and the p53 protein is degraded. So just as a reminder of where you've come across this pathway, this is the p53 slides that you've seen in a previous lecture. Here is p53, here is MDM2. And if you remember back to your p53 lectures, p53 can either be phosphorylated, where it's activated, and if it's phosphorylated it can't be degraded by the proteasome. The reason why is because the corner, as shown on here, the corner of the p53 protein here, when it's phosphorylated, will not dock with the E3 ligase MDM2. So this sort of bit of the molecule here and this bit of the molecule here only interact when the p53 is unphosphorylated. So in the presence of DNA damage, we have unphosphorylated p53, and that can be degraded by MDM2. MDM2 binds to p53, tags on the polyubiquitin chain, and that is degraded at the cytoplasmic proteasome. At the same time, if there's DNA damage present, we can get ATM or ATR phosphorylating MDM2 right on the binding site where p53 binds, and that also prevents MDM2 p53 interaction. So if we go back to this diagram here, if this was p53, then this region here would be the region that is phosphorylated in the presence of DNA damage, and that would destroy or prevent that being a degradation signal. So a phosphorylation on here would prevent p53 interacting with the E3 ligase MDM2, and similarly a phosphorylation of the E3 ligase here will prevent the E3 ligase interacting with unphosphorylated p53. Now, as the ubiquitinated substrate protein comes along and interacts with the proteasome, I previously only shown you the core, which is this bit in sort of orangey pinky colour, but there's also caps on either end, and this is where the ubiquitin receptor and deubiquitinating enzymes will be present. So the ubiquitin receptor will help to bind the substrate. The dubs will then release the ubiquitin for them to be reused, and then the protein can be threaded through the catalytic core, and we end up with our peptide fragments coming out of the bottom of the proteasome. The key point here is that the proteins must be unfolded prior to entry into the proteasome, and they're effectively threaded through. Now, when we get proteins coming out of the other side, they are short peptides, so they're anywhere from a few amino acids up to 20 or 20 plus amino acids. And these are quite important. They can be used, particularly there's immunoproteasomes, which are very much involved in processing peptides for displaying on the cell surface on the on major histocompatibility complexes. So if you're studying immunology, you should be aware that proteins are presented on the cell surface to flag them up to the immune system. And if a cell is infected with let's say a virus, if those viral proteins are degraded and chopped up by the proteasome, they can then be, these short peptides of up to 20 amino acids can be presented on the MHC on the cell surface, and they are then exposed to T cells and other immune cells to identify whether they are foreign or not. So the proteasome is an important component of the immune system. If you're studying immunology, that is very relevant to you. If you are not, then maybe it isn't. Okay, so what I'm going to do is take you through some of the types of ubiquitin ligases and look at some of their specific targets. So there are two major types of E3 ubiquitin ligases. We've got the HECT domain E3 ligases, and I'll come on to next the ring E3 ligases. So the HECT domain ones are homologous to the E6AP carboxy terminus. Now that, for some people, that will mean something to you. It might depend on whether you did my piece of coursework and whether you've studied human papillomavirus, but homologous to the E6AP, some of you may well have written your essays about E6AP. E6 is the human papillomavirus E6 oncogene, oncoprotein, and E6AP is absolutely crucial in allowing human papillomavirus to degrade p53. So this is the family of E3 ligases where MDM2 would sit. We've then got the ring family of ubiquitin ligases. Really interesting new gene is what ring stands for. And all of these E3 ubiquitin ligases have a ring domain in them. Some of the ones that you're likely to come across are SCF, and this is just what it stands for, but it's crucial for the degradation of cyclins as cells transition through the cell cycle. To prevent cyclins from just being randomly degraded, they require phosphorylation of target protein before ubiquitination. So in some situations, a phosphorylation can prevent ubiquitinations. In some situations, a phosphorylation is the signal to be ubiquitinated. And the Anaphase-Promoting Complex is an example of a ring containing E3. So Anaphase-Promoting Complex is crucial for that degradation of Cyclin B prior to anaphase, between metaphase and anaphase, for the metaphase to anaphase transition. So this slide just reminds you about the presence of cyclins and cyclin-dependent kinases in control of the cell cycle, and this is utterly dependent upon the ring family of ubiquitin ligases to control and orchestrate all of these cyclins at the right stage of the cell cycle. So in G1 of the cell cycle we have the G1 S-Cyclin, which is Cyclin E, then we have Cyclin A, which is the S-Cyclin, and Cyclin B, which is the M-Cyclin. As you can see, at each step the cyclins suddenly decrease in amount within the cell, and this is controlled by specific E3 ligases. So here we've got the M-Cyclin, which is Cyclin B levels dropping at metaphase to anaphase transition, and this is controlled by the Anaphase-Promoting Complex. Now, you'll come across the acronym APC multiple times throughout this module. It can stand for Anaphase-Promoting Complex, it could stand for antigen-presenting cell, it could stand for Edematous polyposis coli, which I might talk about in the stem cell signalling lecture, but just be aware APC is used for lots of things, but the Anaphase-Promoting Complex is abbreviated APCC here for the control of metaphase to anaphase transition. So the anaphase-promoting complex is a complex of proteins. We've got the inactive anaphase-promoting complex here. We need activation of a subunit called CDC20, and CDC20 becomes available when the cell is available to undergo metaphase to anaphase transition. That creates the active APC anaphase-promoting complex here, and then that will result in the targeted degradation of the M-Cyclin, also known as Cyclin B. Cyclin B will get a polyubiquitin chain and will be sent for degradation at the proteasome, and that results in the very rapid drop-off of Cyclin B at this stage. The reason why we want the rapid drop-off of Cyclin B at this stage is so that the associated cyclin-dependent kinase CDK1 will then become inactive because CDK1 relies upon Cyclin B for its activity. So when we get all of the chromosomes attached to the mitotic spindle at metaphase and transition through, we would then want to transition through to anaphase. This is when CDC20 becomes available to activate the anaphase-promoting complex and get rid of Cyclin B and then prevent CDK1 from being active. Now, all other parts of the cell cycle, there are other proteins which need to be expressed for a short period of time and then rapidly degraded, and this will include the cyclin-dependent kinase inhibitors such as P21 and P27, because these can inhibit the interaction of cyclins and CDKs and inhibit the cyclin CDK complexes. So if you consider the situation of P21, we know that P21 is induced in response to DNA damage, and that will stop the cell cycle. But once a cell has repaired its DNA damage, then the cell would want to get rid of P21. So to do that, there is the SCF complex, which is one of these ring domain complexes, and this will, when this is activated, will bind to the cyclin-dependent kinase inhibitor, usually once it's been phosphorylated as a tag to suggest that it needs to be degraded, and then we'll get SCF adding the polyubiquitin chain, and then the cyclin-dependent kinase inhibitor will be sent to the proteasome for degradation. So this is a key step that happens once DNA degradation, once DNA damage has been repaired. So continuing the theme of DNA damage and potentially apoptosis, I'm going to look here about the role of the proteasome in regulating specifically apoptosis. So here we're going to look at C-flip. So C-flip is a protein which controls the activation of death receptors. So if you remember back to the apoptosis lectures, TRAIL and Fas ligand and TNF-alpha will bind to their respective death receptors, cause them to trimerize, and in doing so will recruit FAD, and that will recruit in caspase 8. So upon binding of the ligand to the receptor, we recruit caspase 8 to the death receptor complex. The job of C-flip is to prevent caspase 8 binding to this, to regulate, to fine-tune apoptotic signaling. So in situations where the cell wants apoptosis to occur, we will find situations where C-flip will be degraded. So if we think back to the DNA damage pathway that I've just talked about with p53, one of those key proteins is ATM, ATR and CHEK2. They are all kinases which flag up a DNA damage response to various parts of the DNA damage and apoptotic pathways. ATM here activates a protein called ITCH, and ITCH is one of these HECT type E3 ligases. So if ATM is active, ITCH becomes phosphorylated, that becomes activated, and ITCH will then seek out and specifically ubiquitinate C-flip so that C-flip cannot bind in to the death-inducing signaling complex, and that will mean that any death receptor signaling signals will be propagated because caspase-8 will be able to be recruited. In the absence of this, many death receptor signaling signals do not get propagated beyond the death receptor because C-flip will be present and that can block the signaling at this point. So apoptosis is very finely tuned by the proteasome pathway. So here's the whole pathway again, just as a reminder. Here is p53. p53 binds to its own negative regulator, MDM2. MDM2 is an E3 ubiquitin ligase for p53. In the presence of DNA damage, we get activation of CHEK2, ATM and ATR. CHEK2 can phosphorylate p53 so that p53 no longer interacts with MDM2. Equally, we can get phosphorylation of MDM2 so it can no longer interact with p53. If p53 interacts with MDM2, it becomes polyubiquitinated and degraded by the cytoplasmic proteasome. If p53 levels rise because p53 is not being degraded, it then goes on to result in the expression of its target genes, things like BAX, p21 for cell cycle arrest, and the induction of generalized DNA repair enzymes. So the proteasome is crucial in controlling responses to stress, and often stress responses occur very quickly and need to be sorted out very quickly. So DNA damage needs to be sorted quickly, cell cycle arrest needs to be sorted out quickly. What you don't want to do is rely upon the expression of some proteins which then need to be slowly degraded. You need to either activate them or inactivate them very, very quickly, and that's where the proteasome system is really very, very useful. Now there are lots of other stress pathways, and one of those is the unfolded protein response that identifies misfolded proteins. And misfolded proteins are targets for the proteasomal degradation system. Now heat shock proteins, so HSP27, HSP90, also HSP70, these are heat shock proteins, and as the name suggests, if you take cells and you heat them up, you will cause proteins to become misfolded and denatured, and the heat shock proteins are expressed as a response to that to then encourage proteins to refold. So heat shock proteins are not just for heat-induced misfolding, but that is where if you heat up cells, you will induce their expression and stimulate their activity. Proteins like HSP70 binds to exposed hydrophobic patches of proteins. Now if you think about proteins, you tend to get hydrophilic regions on the outside of a protein and hydrophobic regions on the inside, hidden away from water. So that's just how proteins naturally fold up into their most stable conformation. If you find patches of hydrophobic amino acids on the outside of a protein, that suggests that the protein is being misfolded, and proteins like HSP70 come along and tweak them back into the correct conformation. If the HSP system can't refold that protein, then they can recruit E3 ligases to specifically tag the proteins for proteasomal degradation. Now this slide shows how the unfolded protein response can be used pharmacologically, and it also shows the whole process. So misfolded proteins are refolded by the unfolded protein response by chaperone proteins, and those chaperone proteins include the heat shock protein family. So if you think back to the endoplasmic reticulum protein transport lectures, you'll remember that proteins get into the endoplasmic reticulum via a ribosome interacting with the signal recognition particle receptor on the surface of the endoplasmic reticulum. The mRNA is translated and the protein is secreted directly through a translocation channel. Once it gets into the endoplasmic reticulum, it is encouraged to refold because this protein is passing through as a single chain, polypeptide chain. It is encouraged to refold by the chaperone HSP proteins, and here we've got a properly folded protein which can then be sent off for secretion. If the protein misfolds, then that protein needs to have something done with it. Typically, it can be exported out into the cytosol to be degraded by the proteasome system. At the same time, a buildup of misfolded proteins will be signalled by unfolded protein response transducers, and they will signal for either apoptosis to occur if there is chronic unfolded protein response, or if there's a sudden acute creation of an unfolded protein response, that will result in the increased expression of chaperone proteins such as HSP27, HSP90, HSP70. So the presence of unfolded proteins here can either kill the cell or encourage the cell to do something about it. Now this has given an opportunity to use the unfolded protein response and subsequently the proteasomal degradation response as a pharmacological treatment in certain situations. And there is a drug called Velcade, also known as bortezomib, which is a proteasome inhibitor. So if you have a situation where you have lots of misfolded proteins and you block the proteasome, you will seriously stress that cell and it will undergo apoptosis. And this is what the drug Velcade does. It is used specifically in multiple myeloma, which is a B-cell derived plasma cell malignancy. And plasma cells are the professional antibody producing cells. They produce vast amounts of protein, and some of this immunoglobulin protein that they produce, so immunoglobulins are antibodies, as you should know. Some of this immunoglobulin protein is misfolded. If you have cells that produce vast amounts of immunoglobulin that's misfolded and you treat them with Velcade, those cells will undergo apoptosis. Now the reason this is also specifically used for multiple myeloma is because the genetic defect that causes the cancer is usually a defect of the immunoglobulin gene, so that you end up with lots of immunoglobulin protein that is defective in the endoplasmic reticulum that needs to be got rid of somehow. If you can block the way that that protein is degraded by blocking the proteasome, you can stress the cell so that specifically only the myeloma cells will die in response to Velcade. There's another class of drugs which are based around the proteasome, and these are called Protacs, Protalysis-targeting chimeras. And these are drugs that target a specific protein for degradation, exploiting the proteasome. So to develop a Protac, you need to identify a small molecule that binds to an active E3 ligase, identify a ligand for your protein of interest, and somehow link them together so that what we end up with is our target protein being brought in close proximity to an E3 ligase, and then the E3 ligase will then tag that protein for degradation and send it to the proteasome. So this is the way that these drugs are put together. So you've got an E3 ligase. This E3 ligase doesn't have to be the E3 ligase that naturally degrades this protein of interest, but we find a small molecule that naturally binds to the E3 ligase, a small molecule that binds to the protein of interest. We link the two together to create a mature Protac. This means that whenever the E3 ligand binds to an E3 ligase, that will bring the E2 bound ubiquitin in close proximity to the protein of interest. The protein of interest will be ubiquitinated and it will be destroyed. So just looking at what the E3 ligase and E3 ligand are, in some of these Protacs, they're based around thalidomide. So you'll all be aware of thalidomide, which is a drug that has been used, causes some birth defects. The target of the drug thalidomide is a protein called cereblon, and if we take a thalidomide-like molecule, so this is just the derivative of thalidomide, you can use that as the E3 ligase ligand. So this is pomalidomide on here. This will bind to cereblon. We then have a cross-linker, and then we have a small ligand here, and this small ligand can be designed to bind to a protein of interest, which typically is something that is very difficult to drug in other ways. Now RAS, which is a component of the growth factor signaling pathway, you get growth factors binding to the growth factor receptor, that activates the RAS, RAF, MEK, ERK, MAP kinase pathway. This is very, very frequently mutated in cancer, but it's proven to be almost impossible to create a drug that binds to it and degrades it properly. So the plan here is using Protacs is to use pomalidomide to bind, to link a Protac to cereblon, and then use this E3 ligase to degrade RAS by having a small molecule that just binds to RAS, and then we end up with a RAS depleted cell. So this is a completely new pharmacological treatment. Okay, so I've covered quite a lot of stuff here. I'll just do a quick reminder of the key targets of the ubiquitin proteasome system. MDM2 is the E3 ligase for p53. E6, which is derived from human papillomavirus, also does the same thing. It degrades p53. SCF is the E3 ligase for cyclin-dependent kinase inhibitors p21. We've got the Anaphase-Promoting Complex, which is the E3 ligase complex for Cyclin B and Cyclin A. We've got ITCH, which is the E3 ligase for C-flip, and that's activated in response to DNA damage. And then we've got a whole bunch of others. BAX, you remember BAX from the intrinsic apoptotic pathway, that is degraded by a protein called parkin. And the caspases in the apoptotic pathway, on your apoptotic signaling pathways, you'll have seen proteins with the name XIAP, which is the X-linked inhibitor of apoptosis, and CIAP, which is a cellular inhibitor of apoptosis. These are both E3 ligases which result in the specific degradation of active caspases. The job of these is to prevent unnecessary or unwanted apoptotic signaling via the caspase cascade. So you can see that the whole apoptotic cascade and cell cycle are intrinsically linked into the cytoplasmic proteasome and ubiquitination. Okay, there's a brief summary here, and on the rest of the PowerPoint presentation there are some interesting applications of the proteasome system in various aspects of research, such as making green fluorescent protein into a destabilized green fluorescent protein so that it has a very short life, very short survival in the cell, and then we can get do experiments relating to reporter assays with green fluorescent protein. So that's explained on this slide if you're interested in that. So destabilized GFP reporters are explained here. There's a system called the Fucci system, which involves proteasomally degraded cyclin-dependent kinases or cyclins, so that cells are fluorescent in different colours at different stages of the cell cycle. So the Fuji system, follow that link there, which is really interesting system, a really good application of the proteasome system. Something I'm really particularly interested in is the fact that a lot of stem cell populations in cancers have low proteasome activity. This is something I've worked on in the past, but when cells de-differentiate to be more stem cell-like, their proteasome activity is very, very low. So we can identify stem cell-like cells because they will accumulate the destabilized GFP that I talked about here. So if we express destabilized GFP in cancer cells and we see accumulation of destabilized GFP in minority populations of cells, these are the cells which are stem cell-like, because stem cells have a proteasome low phenotype. And there are some complicated pathways which are basically just far more detailed than what I explained in my lectures. Basically everything from up to the summary is required, everything after the summary is just interesting cell biology and biochemistry for you to have a little look at. Okay, I think that's more than enough for today.